Plant tissue culture in one of the vast and advanced and commercially valuable technique to produce plants with desired requirements. There are various techniques to produce traits with required characters, among that culturing through protoplasts is one of the important. First we need to know what is protoplast. Protoplasts are naked plant cells without the cell wall, but they possess plasma membrane and all other cellular components. They represent the functional plant cells such as all cell organelles, but for the lack of the barrier, cell wall. A biological term protoplast was coined by Hunsate in 1880 to refer to the entire cell, excluding the cell wall. If protoplasts from somatic cells of different species can be fused to generate a hybrid, and this process is referred to as somatic hybridization or protoplast fusion. When the protoplast fusion is conducted between the two protoplasts where one protoplast lacks in nucleus is known as cybridization, in simple terms cytoplasmic hybridization, and the resultants are called as cytoplasmic hybrids. Since fusion happened between cytoplasm, these two pictorial representations symbolize the somatic hybridization and cybridization. In order to call is as cybridization, we need one protoplast without nucleus that is enucleated, and that can be done by various methods. There are number of methods to produce cybrids. They are produced in variable frequencies in normal protoplast fusion experiments due to one of the following methods. 1. Fusion of normal protoplast with an enucleated protoplast. The enucleated protoplast can be produced by high-speed centrifugation 20,000-40,000 xg for 60 minutes with 5 to 50% per col. 2. Fusion between a normal protoplast and another protoplast with a non-viable nucleus or suppressed nucleus. 3. Elimination of one of the nuclei after heterokaryons formation. 4. Selective elimination of chromosomes at a later stage. 5. Irradiating, with X-rays or gamma rays, the protoplasts of one species prior to fusion in order to inactivate their nuclei. 6. By preparing enucleate protoplasts, cytoplasts, of one species and fusing them with normal protoplasts of the other species. Isolation of protoplast. For somatic hybridization or cybridization, the protoplast need to be isolated from plant tissue that may be leaf, stem, root or either from any part of the plant. The first isolation of protoplasts was achieved by Clarker, 1892, employing a mechanical method. The removal of cell wall from the plant cell is called as protoplast isolation. And 2 techniques. 1. Mechanical method. 2. Enzymatic method. Mechanical method of protoplast isolation. Protoplast isolation by mechanical method is a crude and tedious procedure. This results in the isolation of a very small number of protoplasts. The technique involves the following stages. A small piece of epidermis from a plant is selected. Then cells are subjected to plasmolysis where protoplasts gets to shrink away from the cell walls. Then tissue is dissected to release the protoplasts. Mechanical method for protoplast isolation is no more in use because of the following limitations. High yield of protoplasts and their viability is low. 2. It is restricted to certain tissues with vacuolated cells. 3. The method is laborious and tedious. However, some workers prefer mechanical methods if the cell wall degrading enzymes of enzymatic method cause deleterious effects to protoplasts. Enzymatic method of protoplast isolation. Enzymatic method is a very widely used technique for the isolation of protoplasts. Because of advantages such as good yield of viable cells and minimal or no damage to the protoplasts. Enzymes for protoplast isolation. The enzymes that can digest the cell walls are required for protoplast isolation. Chemically, the plant cell wall is mainly composed of cellulose, hemicellulose and pectin, which can be respectively degraded by the enzymes cellulose, hemicellulose and pectinase. The enzymatic isolation of protoplasts can be carried out by two approaches. 1. Two-step or sequential method. The tissue is first treated with pectinase, macerozyme, to separate cells by degrading middle lamella. Then these free cells are then exposed to cellulase to release protoplasts. Pectinase breaks up the cell aggregates into individual cells, while cellulose removes the cell wall proper. 2. One-step or simultaneous method. This is the preferred method for protoplast isolation. It involves the simultaneous use of both the enzymes, macerozyme and cellulase. The isolated protoplasts are then subjected to culture, which is called as protoplast culture, by providing suitable culturing conditions. Upon appropriate conditions, cell wall gets regenerated around protoplasts and undergo successive division to form a mass of undifferentiated cells known as callus, which then gets differentiated into tissues. Once protoplasts were successfully isolated, it's important to check their viability before going further. Viability of protoplasts. It is essential to ensure that the isolated protoplasts are healthy and viable so that they are capable of undergoing sustained cell divisions and regeneration. There are several methods to assess the protoplast viability. 1. Fluorescein diacetate, FDA, staining method. The dye accumulates inside viable protoplasts, which can be detected by fluorescence microscopy. 2. Phenyl stain is selectively taken up by dead protoplasts, turn red, while the viable cells remain unstained. 3. Exclusion of Evans blue dye by intact membranes. 4. Measurement of cell wall formation, chalcofluor white, CFW, stain binds to the newly formed cell walls which emit fluorescence. 5. Oxygen uptake by protoplasts can be measured by oxygen electrode. 6. Photosynthetic activity of protoplasts. Once viability of the protoplast was completed, then the protoplast were need to be fused for hybridization, and that can be done by two types. Method number 1. Spontaneous fusion. Protoplasts during isolation often fuse spontaneously, and this phenomenon is called spontaneous fusion. Simply physical contact is sufficient to bring about the spontaneous fusion among the similar parental protoplasts. Method number 2. Induced fusion. Fusion of freely isolated protoplasts from different sources with the help of fusion-inducing chemical agents is known as induced fusion. Normally, isolated protoplasts do not fuse with each other because the surface of the isolated protoplast carries negative charge around the outside of plasma membrane, and thus there is a strong tendency for protoplasts to repel one another due to their same charges. At that time, external induction need to be done by using inducers such as chemicals, electrical shocks, etc. Chemofusion. Several chemicals have been used to induce protoplast fusion and called as chemofusion. Sodium nitrate, NaN03, polyethylene glycol, PEG, calcium ions, Ca2+, polyvinyl alcohol, etc. are the most commonly used protoplast fusion-inducing agents, which are commonly known as chemical fusogens. Chemical fusogens cause the isolated protoplasts to adhere to one another and leads to tight agglutination followed by fusion of protoplast. The adhesion of isolated protoplast takes place either due to reduction of negative charges of protoplast or due to attraction of protoplast by electrostatic forces caused by chemical fusogens. I. Fusion by means of nano 3. It was first demonstrated by Kuster in 1909 that the hypotonic solution of nano 3 induces fusion of isolated protoplast forming heterokaryo hybrid. This method was fully described by Evans and Cocking, 1975. However, this method has a limitation of generating few no of hybrids, especially when highly vacuolated mesophyll protoplasts are involved. 2. High pH and Ca++ treatment. This technique lead to the development of intra- and interspecific hybrids. It was demonstrated by Keller and Melcher in 1973. The isolated protoplasts from two plant species are incubated in 0.4 m mannitol solution containing high Ca++, 50 mm CaCl2.2H2O, with highly alkaline pH of 10.5 at 37 degrees Celsius for about 30 minutes. Aggregation of protoplasts takes place at once and fusion occurs within 10 minutes. 3. Polyethylene glycol treatment. Polyethylene glycol, PEG, is the most popularly known fusogen due to ability of forming high-frequency, binucleate heterokaryons with low cytotoxicity. With PEG, the aggregation occurred mostly between 2 to 3 protoplasts, unlike Ca++-induced fusion, which involves large clump formation. Electrofusion. Recently, mild electrical stimulation is being used to fuse protoplasts. This technique is known as electrofusion of protoplasts. In this method, two glass capillary microelectrodes are placed in the protoplast's suspended solution. An electrical field of low strength of about 10 kV gives rise to the electrophoretic dipole generation within the protoplast suspension. This leads to pearl chain arrangement of protoplasts. The number of protoplasts within the pearl chain depends upon the population density of the protoplast and the distance between the electrode. Subsequent application of high-intensity electric impulse of 100 kV for some microseconds results in the electric breakdown of membrane and subsequent fusion.